for really any disease types, specifically colorectal cancer, as we're trying to figure out, is it one big disease or is it multiple little diseases based on molecular subtyping? Are you a lumper or a splitter as a history major would be what we would call that. And then why it takes so damn much time and money to answer what seems to be a relatively simple question. Does drug X work? The fact that that is such a hard question to answer is partly why we're all here together trying to figure this out. So why does it matter? Currently, if you look at different publications, this one is from the Journal of Clinical Oncology. It's estimated to cost about $1.2 billion to successfully bring a compound from design to market. And most drugs fail. The number of sort of compounds that start off with potentially therapeutic intent that end up actually being approved by the FDA for a specific indication is on the order of 2%. And that's probably an overly optimistic estimate. The cost of failure in this wilderness is incredibly high, both in terms of finances, but also in terms of patients. If you have a drug that fails, that ultimately has no benefit, what happened to all of the patients who were exposed to that drug? Hopefully it wasn't harmful, but we don't know. So it takes about 8 years for a drug, I'm not even talking about the preclinical development. From an idea to phase 1 probably takes a decade. But from a phase 1 trial, which I'll explain in a second, to actual FDA approval takes about 8 years. There are some exceptions, there's some fast tracking that we'll talk about. Now, so Otto von Bismarck is not often included in academic discussions, but he had a great line which was, you never want to see laws or sausage get made. Right? I would also argue that clinical trials and medical science is also a process that's sort of dirty and a little sausage-like. You don't really want to know what's in it. You want to know the end result. So how do we decide what works? This is a messy process. It starts with an idea. It starts with Dusty in his lab with some spheroids, squirting stuff on them and saying, hey, do these make the cells die? And then we do this high throughput screening. So if we have access to dozens or hundreds of compounds, we have cells in a dish, we can start to analyze through high throughput screening, basically a very fancy way of testing a bunch of in vitro cells all at once. What makes them die? What makes those cells go away? And then animal models are a little bit better because then it starts taking some of the variability of normal tissue in play. So Dusty's animal models are really important things, as are the sort of spheroid cultures, trying to figure out how these cancers behave not just in a dish, but in a living thing. We can't realistically do these experiments on humans. The risk is too great and you'll see how some of the experiments that we do in phase 1 clinical trials are scary and potentially very unsafe. But at least in that process of informed consent, we can explain a little bit better about what that is. We have tons of improvements in the area of animal modeling so we can do more preclinical work before we expose patients to potentially toxic compounds, but we have a ton more work to do. So these are my 3 children. Ben is 5, John is 4, and Mary is 1. And if I want them to do anything or learn anything, it has to be in the form of a story. So I have to tell you guys a little bit of a story to walk through this. Ripped from the headlines, right? We all were around when Law and Order was on. And in fact, right around the time that Law and Order was a show in 1990, I actually had to look that up, was around the time that the inhibition of epidermal growth factor receptor was found to be possible. So in a lab in Israel, this compound got synthesized. Now, there is a ton of the backstory behind this drug that became cetuximab. And who's had cetuximab in the room? Anybody? Okay, so how this drug got developed is a sausage. Okay, this is totally weird and disgusting as to how this thing came from this lab in Israel to people, to actual benefit. And I think this is particularly illustrative of the process. The 2 people who were in this lab actually got into a very nasty intellectual property lawsuit. as to who actually came up with it. So the guy who came up with the idea of blocking EGFR called this other guy and said, do you have any compounds that block this pathway? And so that was where the fight came in, and you'll see why. So phase 1 clinical trials. Once you have the idea of a pathway and you have a drug to hit a pathway, you have to figure out how do I give that drug to people? Phase 1 clinical trials are dose finding. There is no endpoint of efficacy in a phase 1 clinical trial. There is no expectation of improved survival in a phase 1 clinical trial. That's really scary. But it has to be included in the consent process for patients to say, I am giving you access to this drug. It works in a dish. We don't know the dose or the schedule. That's why we're doing this trial. So the way it starts is at what's called the LD10, the lethal dose for 10% of the animals. Yikes. That's the starting dose, and then we escalate up from there. That's really scary. However, this is the kind of thing that we believe to be safe and why the phase 1 trial is necessary to get to escalating to a dose that we believe to be therapeutically effective. So a traditional phase 1 study is for patients with refractory solid tumors, a very unselected population, people with cancer that has progressed through stuff that's available. However, there are folks who are still feeling okay after that process and really giving of themselves. I really think my patients who are on phase 1 trials give such a tremendous gift to the rest of us, all of us, that I am willing to stand up and say, go ahead, try it on me. It is a high risk, high reward for patients because the potential reward is if you're in that lucky percent where it works, there is nobody else in the world who can get access to this compound. Many of these compounds are known by a series of numbers and letters. IMC225 in the case of cetuximab. MK3475 for Pembro, is that what it was? Yeah. So, and you'll often hear, by the way, particularly nerdy oncologists use the numbers and letters to be like, I was there, it was still VP16, you know, like, it's like a band and hipster thing. So the endpoints are really toxicity. When do people have a significant dose-limiting toxicity? When does the rash get so bad that we can't go higher on the dose? When do they have bone marrow toxicity? Or, God forbid, when does somebody die? And so this is all open label. There is no randomization. There is no placebo arm. This is, we know what you're getting. We think this is the right schedule to give it. And that's how a phase 1 trial works. Classical 3+3 design is basically you enroll 3 patients at a time at a dose level and see if they have toxic side effects. If one patient has a toxic side effect, you expand the cohort to another 3. And if another patient has a toxic side effect, you have basically a third chance of having something very significant happen. That's the maximum tolerated dose. If, however, you have no toxic responses, you can go up to the next dose, and then you keep going, following this algorithm to get to what's called the maximum tolerated dose. Does that make sense? Mm-hmm. So it is really it is a true experiment in the truest form of saying, I'm going to give this to you and see what happens. What does success look like in a phase 1 clinical trial? Really, success looks like you have a dose that's biologically plausible. In the in vitro studies or in the mouse models, what was the dose that it took to make a tumor do something that made you want to test it? Can we get to those levels in people? And what happens to them when we get there? Is it biologically plausible to move this drug down the road? Did anybody respond? And the crazy part is, a historical response rate for a phase 1 trial is 5%. So almost every center looks back at their phase 1 experience and says, how many responses have we had over the last decade? Almost every academic center that has a phase 1 program has a response rate of around 5%. One of the key things that we want to look for is, how can we design trials better and pick patients better for these targeted therapies to improve that response rate? Okay, so what happened with cetuximab? So IMC225 showed 2 responses in 13 patients. That was enough to move on and to get published in our big journal, Journal of Clinical Oncology. The swimsuit edition of that is terrible. So based on the responses that were shown in that clinical trial, because Dusty and I are in it, it's not that good. But 2 responses out of 13 was enough to have, sort of a slow clap at a national meeting and say, hey, let's expand this cohort to head and neck cancer and colorectal cancer. So now that we have a dose and a schedule, how do we determine success? We look for objective response rates. We look for tumor shrinkage on scans. That requires more patients. We can't just do this with 13 patients. So a typical phase 2 trial is somewhere between 40 and 100 patients. It is very often open label with no randomization. Sometimes we give it with existing therapy. Sometimes we do randomize. A randomized phase 2 trial has the drug that you're interested in plus either a control arm of what's already out there or the drug you're interested in plus another drug. Does that make sense? These still are typically late-line, heavily pretreated patients. A randomized phase 2 trial in the first-line setting is a dangerous thing because you don't know how well it works. And if you're moving it in front of stuff that we already know works, that's a very dangerous proposition, and you better be double damn sure that that's going to do something. So the endpoints that we look at for a phase 2 trial are response rates. What percentage of patients have tumor shrinkage? Progression-free survival. How long does it take for patients to have their tumors grow beyond a certain threshold? Or a disease control rate. How many patients have stable disease? So a home run, and since it's baseball season, I want to put Carlton Fisk back up there. So a home run would be, in a heavily pretreated population, having anything more than a few percent of patients have tumors shrink. And so what that means is, if you have a real home run where everybody responds, you can get accelerated approval from the FDA through a phase 2 study. That is vanishingly rare, but possible. So then after you have a successful phase 2 study, you can start thinking about other trials to get this drug incorporated into clinical practice. Now, in a heavily pretreated population, 9% of patients with EGFR overexpressing colon cancer responded. That's a big deal. There was tons of excitement in the community. 60 Minutes did a piece on it in 2001 advocating for all colorectal cancer patients to get access to cetuximab in 2001. And this was thought to get FDA approval on a fast-track basis. Tons of people investing in a company called ImClone, which is what IMC 225 made. So again, another real exciting, lots of enthusiasm for that. However, the confirmatory studies weren't there. FDA approval was held up. Investors in ImClone lost a lot of money. Martha Stewart went to jail for cetuximab. I forget, Dusty, do you remember this story? I think it was her son-in-law or something like that was an executive at ImClone and leaked to her the fact that the FDA was not going to approve cetuximab, so she sold. 3 days later, the FDA said, we're not approving this drug. Stocks of ImClone crashed. And remarkably, however, and so that's why Martha went to jail. That was the insider trading from ImClone. The only thing that ImClone made was cetuximab. That was its only revenue stream. Remarkably, however, the drug didn't die then because it still had an indication in head and neck cancer, and it still in colon cancer worked in a subset of people. Why did it work in that subset of people? And that's where the science comes in. In the 5 patients who responded, what was unique about them? Why didn't the other 52 respond? So really understanding this pathway, how blocking the epidermal growth factor leads to a signaling cascade that says cancer cell grow. What we learned, and I think something that most of us are familiar with now, is we can block up here till the cows come home, but if this KRAS mutation is present or a BRAF mutation is present, you can block up here all day, but this signal is still going to go to the nucleus to say, divide cancer cell. Does that make sense? Yeah. So once that was teased out, they went back to the drawing board to say, hey, maybe we can use this not just in EGFR overexpressing cancers, but in folks who are absent a RAS or a RAF mutation. So that's where the next set of studies came through, and it actually showed a survival benefit in pretreated patients. And the second-line therapy, cetuximab, got FDA approval in 2004. Now, there's a sign outside of Dusty's lab. One of his lab techs has, "Do all the things." So she's a very enthusiastic person. And this really underscores what happens with a drug that gets approved. Now it's like, let's test it for everything. Let's look for every possible avenue in which we can use this drug. Not only from an altruistic perspective, is there patient outcomes on the line? There is a ton of money on the line. And for us, the patient outcomes mean something, but for the people who invested $800 million in developing the drug, they want to reap some of that benefit. So then we have to go to phase 3 studies. So after showing safety and efficacy, phase 1, safety, phase 2, efficacy, now phase 3 has to show improvements when compared to that which is already out there. Does that make sense? So that takes hundreds of patients, and that requires blinding and randomization. We can't know who's getting drug X. Because if we knew that, and as investigators we had some implicit bias to say, I want drug X to work, it's not a failing more than it is just us being humans. We want these things to work. So to avoid bias, it takes blinding and randomization. Being on a phase 3 trial doesn't mean you get exposure to drug X. This is a high-risk, high-reward proposition, not for the patients really anymore, because the safety and efficacy have already been worked out. The risk-reward proposition now is for companies, because this requires millions of dollars of investment. Requires a ton of regulatory oversight. The institutional review board, is this safe? Is this ethical? Data safety monitoring, who is responsible for deciding what a response is or isn't? And it has to be protocol driven. We have to be able to treat patients the same way here that they will in DC, that they will in Colorado, that they will in Seattle, that they will in New York. Does that make sense? So creating the recipe that goes in the cookbook. Typically this is at least sponsored by companies, but a real gold standard is when it's done by a neutral party. Having a governmental institution run this with no skin in the game other than the well-being of the populace. So if you see a phase 3 clinical trial that is entirely funded by a company, there is some skepticism that creeps in there. So the design options, we have to first design who is the population. The drug cetuximab failed when we took all patients with colon cancer. It succeeded when we said patients with KRAS wild type tumors with no mutation that will say it's not going to work. And then we have sort of a control arm, which would be standard chemotherapy. And the treatment arm, which is chemotherapy plus cetuximab. Makes sense. And then the other questions are, is the randomization ethical? Do we do a 2-to-1 randomization to get more people access to the drug? Complete the study faster, but it costs more money to do that. Do we offer crossover? If it works, for the people who were on the placebo arm, do they get access to the drug at the conclusion of the study? Wow, so some very complicated questions go into clinical trial design. So the brass tacks endpoints are, does this drug make people live longer? I don't care as much if it makes studies prettier, if it makes a CAT scan look nicer down the road. I want to know, is this going to make my patient live longer? There's a ton of safety data because in this large population, this is where the rare side effects come out. So all these commercials about talk to your doctor about, those all come out of phase 3 clinical trials. The side effects that were reported in the phase 3 clinical trials are the ones that get reported on the commercials with the bathtubs. P.S. The fact that we're spending billions of dollars a year on that particular medication versus cancer drives me nuts. The fact that I can't watch an NFL game without my 5-year-old saying, Daddy, what's erectile dysfunction? Crocodile that is ineffective. That's all it is. To complete the story, and sadly it's true, he's in kindergarten and it's like, oh, okay, Benny. So phase 3 clinical trial, these are examples of phase 3 clinical trials for cetuximab. So Crystal, FOLFIRI plus cetuximab in KRAS wild type patients. Response rate's 59.3%, so a 10% improvement over FOLFIRI alone. Wait a minute, I thought this was a home run drug. We're talking about response rates that are only a little bit better. overall survival improvement of 3 months. Is that really a home run? Well, I kind of think so. Coin, response rates improved, but overall survival wasn't really changed. Bond 2, using cetuximab plus Avastin. Anybody take Avastin? Have you been on Avastin? Yeah, okay. So using cetuximab and Avastin in combination, response rates less, toxicity higher, death sooner.
Yeah.
What? So using these two drugs that thought, there was an editorial in 2000 about Avastin saying the end of cancer, the magic bullet, right?
Saved my life.
Yeah. Well, and Avastin's a hell of a good drug. Yeah. But when used in combination with other drugs, it doesn't play well. And then this study, the adjuvant study, PEDAC 8, addition of cetuximab did not improve disease-free survival. Now, look at all of these patients. So I did some math. If it costs $25,000 per patient to be on a clinical trial, which is a pretty reasonable number for all the regulatory oversight and all the process, 6,142 patients in these 4 studies. Now, there were dozens more. But this, conservatively, is an outlay of $153 million for those 4 studies. And, so high risk, high reward proposition. So the design of trials means something. And then throw into this that around 2004, 2005, Amgen came out with panitumumab, a humanized version of cetuximab. Dosing it every other week costs 2 to 3 times as much. Similarly confusing results were seen. Improvements in survival in some studies, not in others. So after all that, thousands of patients exposed to this drug. Eli Lilly bought ImClone and its only compound, which was cetuximab, and then what turned into ramucirumab, which is now in clinical trials, for $6.5 billion. Holy crap. So what did we really learn? That targeting EGFR is possible, safe, and does improve overall survival in patients with metastatic colon cancer, as long as they don't have a mutation in KRAS. So there were some surprising findings that why didn't it work in the adjuvant setting? Why didn't it work in the no evidence of disease setting? We still don't know that answer. So we have learned that response rates are better with cetuximab the sooner we use it. However, we don't have a significant overall survival change relative to whether we start with Avastin or cetuximab. There are other mutations besides KRAS that mean something, all of these other RASs, and responses are great things, but they're not the cure. The drug works for a long time, but it's not going to eradicate the cancer from people's bodies. All right, so why does that matter? I think when you guys are out there reviewing studies as patient advocates and things like that, you have to ask, is the question valid? Will the study answer the question? And is the study worth the risk to patients? Is it worth the risk to society? You don't really need to care if it's worth it to the company. The company's already saying, we're going to fund this. But the bigger question is, do we benefit from a scarce resource? Do enough patients benefit from this to put those 6,000 or whatever patients potentially in harm's way? So these questions are critically important. And the slides, by the way, I sent out so you can get those. You don't need no slide. So cooperative group studies are critical. We don't have 6,000 patients with colon cancer that walk through the Carbone Cancer Center anymore. We couldn't accomplish this study alone. So ECOG Akron is one group that we're a part of. We helped found it here, actually. So both academic and private practice groups around the country are members of ECOG who can enroll patients on these trials to increase the N. SWOG is another group, Southwestern Oncology Group. There's the new Big Ten Cancer Research Consortium. So we're really trying to get that up and off the ground to improve our numbers, to get more people exposure to the science that we do. Cooperative groups have great advantages. You can get access to tons of patients, and once the sites are open, you can accrue relatively quickly. The investigators themselves are typically well respected. Being at the university of something you've heard of gives you a chance to present the data and say, oh yeah, this is Mike Bassetti's study. He's not some schlock. I know him. He's devastatingly handsome too. He just walked in the door. Leveraging the strength of different institutions, saying, hey look, Michigan has great radiation oncology. Wisconsin has great radiology and medical oncology. How can we bring these strengths together? So maybe they bring radiation oncology techniques from Michigan, maybe we do a central review of the imaging studies here. So we can take the best of each institution in a cooperative group and put it together. And we can ensure protocol adherence. If everybody's reading from the same playbook, written the same way, with the same group reviewing the data, we can say this is a standard population. This reflects the population as a whole. Does that make sense? So disadvantages. This is slow to design and open. Sometimes practice isn't uniform. Sometimes people have their way of doing things. God forbid oncologists be sort of stiff-necked group of people. And it takes a ton of staffing to get it right. I'm glad Renee just walked in when I can say that it takes a ton of people to get a clinical trial up and running the right way. So how it works, this is really where the sausage part comes through as well, that ideas from investigators go through a steering committee, a national body of esteemed clinicians to say, this is how we want to do this. And then the steering committee says, yeah, we want to move this forward because this is an outlay of time, money, and patience. And to have it sort of willy-nilly, to have somebody just throw an idea out there and see if it sticks, there has to be more to it than that. The idea needs a sponsor. Who's going to supply the drug? Who's going to pay for the actual science? Who's going to help Dusty's lab do more mouse colonoscopies? And furthermore, who's going to help make sure that the data is safe, secure, and that the study's done in a patient-centered and safe way? So once the go-ahead is achieved, it seems like a no-brainer that everybody involved in the cooperative group wants to be involved. But sometimes, well, you know what? We don't have enough of that population, so we're not going to open it here or there. And then once accrual is complete, the findings are presented and published. Natalia and I get job security because we've presented this great study, and now we get to pay for my season tickets for the badges. What about this moonshot thing? What is the Cancer Moonshot Initiative? So a lot of it has to do, and I wanted to put this slide from The Onion because I just love how they've turned Joe Biden into like everybody's creepy uncle. He's really not that. Right? He's not the guy washing the Trans Am in the front yard of the White House, but it's sort of an awesome picture. The idea behind the moonshot that Joe is pioneering, largely because of his son's death from cancer, is to try to decrease the burdens of infrastructure. To try to say, look, if somebody has a really good idea, how can we move that faster? How can we get that further down the line without all the administrative hurdles while still being safe? So I think stay tuned for that, because I think the goal is to make our discoveries faster at a lower cost. But the hard part is, this is complicated stuff, right? I mean, this is just one set of pathways. There are dozens more that play into oncogenesis. So that's why it takes all these clinical trials to answer these seemingly simple questions of, does this drug work? I'll stop there. And the sad part is, the reason I have to leave is because I have to go to a funeral. One of my patients who had esophageal cancer for 6 years, after all of that, he died. And I want to stop wearing black suits. I don't want to do this anymore. So anything we can do to keep that from happening, I'm on board.
Sam, when does a patient know that it's a good time to go on a clinical trial?
I think a couple of points. One is when you start going off a paved road, right? For colon cancer, we have a pretty good paved road of either FOLFOX, then FOLFIRI, or FOLFIRI, then FOLFOX. And then mutational status will help dictate either cetuximab or Avastin. And right now, I mean, those drugs were pioneered and presented in 2004. We have not had anything come close to beating that in 12 years. So when there's something that has generated a lot of enthusiasm in an early phase, there's a buzz about it, like there was with cetuximab early on. And so when folks are running low on options, start thinking about, okay, first-line therapy failed. Anytime therapy fails, it's time to look around for all options. Standard therapy, clinical trials. At initial diagnosis, same thing. Yeah.
Quick question. If accrual takes so long for some clinical trials, and you get a patient who joins that trial early on versus the patient who joins the trial a year into the accrual, doesn't that first patient get more of an advantage of taking that? I mean,
what if in the dose escalation, the first patient gets a dose that's subtherapeutic?
I don't know.
Yeah, so I think being...
So doesn't that skew results?
No, and I think that's why they're protocol driven. We can't, what becomes a challenge in clinical trial enrollment is if that first patient does really well and 60 Minutes is out there with a camera saying, look at how good this person did, everybody else is gonna say, forget the trial. Where can I buy the drug? Where can I get this off label? And then the trial can't accrue because people are getting this stuff from other sources. That's why it's so important to have a trial and stick to it. And in our practice, we try to really minimize off-label use as much as we can. And why Dusty's molecular tumor board is such an important thing to say, if we're doing that, we better have a damn good reason why, and we better be studying the outcomes. Does that make sense?
Yeah.
So if you're the first patient on the home run drug, you will ultimately get the same benefit because the dose isn't going to be all that much different. But what may end up happening is other patients may not be able to get on study if it works too well.
Or if other patients don't have that success, how does that, I mean?
Well, then we start figuring out why did it work or why didn't it.
Okay, thank you.
So to what extent does competitiveness among these various research centers and the desire to monetize the discoveries impede the formation of these cooperative groups and the operation of these cooperative groups?
Probably right. And I think the nice part is working at a state hospital that is entitled not-for-profit means that we don't actually get financial benefit from the trials that we run.
Well, I was at a meeting where Johns Hopkins was doing the presentation, and two of the Johns Hopkins people had just left Johns Hopkins to form a company to commercialize something that they had discovered while they're at Johns Hopkins. So
yeah, absolutely. I mean, I think in that sense, that building right there is the Wisconsin Alumni Research Foundation, Warfarin, Wisconsin Alumni Research Foundation, Erin. That's where it got discovered, and that's how it got monetized. 5 cents a pill helped build that hospital one brick at a time. So, and that's another cool story, but I don't have time. But the point of it is, yeah, if somebody makes the initial discovery, those two scientists in Israel got in a multi-million-dollar lawsuit because there were billions on the line.
because it seems to me there are so many clinical trials going on, that there has to be a lot of duplication, and even a lot of duplication of lab studies before you get to clinical trials. And wouldn't it be great if there was some way to, make all that known? I mean, for you to present at ASCO or to write a paper for a journal takes a lot of time.
Yep.
Even to get accepted.
Yep.
And so how widely disseminated all those results, especially failures.
Totally right, exactly. And I think part of it is that the investigators are directly invested in, I think a truly altruistic way, I hope, the success and failure of their ideas and their patients, not, I want this to work because I'm stubborn. No.